The overall goal of this procedure is to compare cell wall polymer composition across a broad range of plants or tissue types using glycan microarrays. First, harvest the plant tissues of interest. After homogenization, prepare alcohol-insoluble residues, then extract the cell wall glycans sequentially using CDTA and sodium hydroxide. Then, print the cell wall glycan extracts in a microarray using a robot. Probe the microarrays with monoclonal antibodies directed to cell wall glycans and quantify the spot signals on the microarrays. Ultimately, results can be used to determine the relative abundance of glycan epitopes across a sample set of interest.
We developed this microarray method when we recognised the need to provide a global snapshot of cell wall glycan occurrence across multiple plant tissue types simultaneously and detect compositional differences.
So the main advantage of this technique over traditional methods such as monosaccharide analysis is that this is a microarray-based platform where hundreds of samples can be screened simultaneously. This is a rapid and comprehensive method of profiling cell wall composition.
For each tissue of interest, start with triplicate samples of 100 milligrams fresh weight of plant tissues. Using liquid nitrogen, homogenize the samples to a fine powder. Then, transfer the homogenates into 10 milliliter plastic conical